We spent a lot of time leveraging standard H&E based samples in order to do our AI-driven digital pathology. And in this case, we were able to leverage what's called an IHC or immunohistochemistry stain, particularly for an immune cell biomarker called CD8, which is a marker of active lymphocytes that are able to infiltrate into the tumor microenvironment and leverage an immune response to tumor presence or cancer growth. We trained a deep learning algorithm that was able to detect all nucleated cells within the tumor microenvironment and specifically classify CD8 positive and all other cells based on their morphology. On top of that, we were able to layer an extra biomarker analysis, if you will, which is called immune phenotyping. In this case, CD8-based immune phenotyping, where we can train another kind of classifier. In this case, we tried two different independent approaches in order to classify the slide level immune status of these patient samples as either immune inflamed, immune excluded, or immune desert. At the onset of this project, we weighed whether or not we should stick to a single approach, either approach 1, which is the cutoff-based approach, or approach 2, which is the spatial-based approach. However, we realized that we had a great opportunity to try both and actually do a head-to-head comparison and see whether one outperformed the other. So this first approach, the cutoff-based approach, relies specifically on two key features, which are our CD8 positivity features. So CD8 lymphocyte proportion within cancer parenchyma and within cancer stroma. You're only using those two features. So that's also a con of this approach. So while it's highly interpretable because you know exactly what this classifier is learning from, you're only taking into account a few features, despite the fact that we have many, many more rich quantitative features that we're able to extract from these images. The other approach, the spatial-based approach, is pretty different. So what it does is it actually takes into account all of the predictions we're able to make within the tumor microenvironment, including CD8 positivity in lymphocytes as well as other cells and where they are located. So that's a huge pro of this approach, is that you're taking into account all of this spatial contextual information. However, the con of the spatial approach is that it's just less interpretable by virtue of the kind of classifier that you're able to train, which is called a graph neural network, or a GNN. Both approaches were comparable to an average annotator's ability to classify these samples in comparison to a consensus of expert pathologists. So that established as a proof of concept that both of these approaches perform quite similarly to each other, which was a key result. The clinical utility of having classifiers that can predict immune status is the scalability and the ability to deploy these methods across potentially thousands of patient samples in order to do in-depth outcomes-based biomarker analysis, hopefully to identify patient subpopulations that are more likely to benefit from particular drug therapies.